Hello everyone, welcome back. This is chapter 8, the immune system. I'm going to go ahead and preemptively call this part 1. We'll see if it actually is part 1. I don't know. I don't think we'll get through all of it because we do have to cover a little bit from the cardiovascular chapter as well, just going over like the components of blood and things like that. So I think that's a good place to start as anywhere. If you'd like to support the channel and everything that we do here, check out the links in the description. 50% of everything you donate goes to Doctors Without Borders. Let's get started. Blood. We've talked about red blood cells. Now let's talk about white blood cells. Red blood cells are the oxygen-carrying cells inside of your blood, whereas white blood cells are sort of just all hands. Just all hands all the time for anyone and anything. They want to fight everyone. They want to fight every single possible thing in the world. Red blood cells were known as erythrocytes. What are white blood cells known as? Leukocytes. Don't confuse that with lymphocytes, which are a type of white blood cell. These are leukocytes. And leukocytes actually comprise less than 1% of your blood volume. Very little. And it can increase in infection and things like that and blah, blah, whatever it is. And they divide into two classes. There are granulocytes and granulomites. No, agranulocytes. Sometimes I just like to lie to you guys because you get so scared and you're like, that's not what I read. Because you guys are nerds who read ahead. As you should be doing, by the way. You should be very far ahead of me. And if you're not and you're planning to take the MCAT in March, hello? Pick up the pace. There's someone in the room who's not in their third year of college. I'm not going to single them out, but it's quite early. But I don't mind their plan. The plan's not bad. Granulocytes include neutrophils, eosinophils, and basophils. I will never write this entire thing out ever again. This is N phi, E phi, and B phi. And these contain cytotoxic granules that modulate inflammatory reactions, allergies, specific pathogen recognition, or specific pathogen attack. You will learn in medical school that eosinophils are very specific for parasitic infections, right? Etc. So they can attack bacteria, they can attack parasites, they can do blah, 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 this is that. Agranulocytes include lymphocytes and monocytes. Monocytes are like your macrophages, things that ingest other things. They're named different things in different organs, microglia, Langerhans cells. And actually, remember when we talked about the osteoclasts, when we talked about parathyroid hormone, how they chew on the bone? Those are macrophages. Osteoclasts are macrophages, right? Very specialized macrophages. Lymphocytes are B and T cells, which regulate specific immune response. I hate immunology with a burning passion, with a fiery, burning passion. And the reason for that is because this is bad enough. And then you get to medical school. And it's IL-1 through 13 and TNF-alpha and all these signaling pathways, and this one turns on this one, and the CD40 ligand interaction over the blah, blah. I don't care, man. I don't care. You just give them Tylenol. Like, oh, you have a flesh-eating bacteria? Here's some Tylenol. Yeah.
What's the last word after specifics?
Pathogen attack. Yeah, so bacteria and parasites, things like that. Right. And then the final thing that we have in blood are platelets. Platelets are cell fragments. So there's a cell called a megakaryocyte. It explodes and makes a bunch of platelets. That's it. We talked about blood antigens last time, right? A blood groups, B blood groups, O blood groups, things like that. We did that in cardiology, right? So let's talk about the structure of the immune system. So these are the blood cells. You guys done with that? You guys all right? Yeah? Anyone still writing? You were. I should honestly just like hot mic you guys. I should take the second microphone and you guys just like toss it around. And everyone gives their 2 cents like, oh my God, I'm drowning. Someone please send help. We have 2 types of immunity. Innate immunity. And adaptive immunity. One of our teachers at Qalam is obsessed with charts. So every single time I draw out a chart, I think of him. Everything he does is in a chart. I'm pretty sure he eats breakfast in a chart. Like, he's awesome. He's a great teacher. Fantastic teacher. Innate immunity can fight pathogens non-specifically. This is a free-for-all. You're a pathogen, you're getting it. You're getting cooked to the best of my capacity, right? But let's say that you built gloves specifically for boxing, right? And then you just use regular like gloves that you wear for the winter that keep your hands warm. Obviously, the ones that are made specifically for boxing are going to be better at boxing. And the ones that are made to keep your hands warm are just gloves, right? This is like wearing a glove. This is like wearing a boxer's glove. This might work in protecting your hand and might pack a little bit of an extra punch. This is going to murder the guy, right? So that's the difference that we're going to see here. So the adaptive immunity, also known as specific immunity, is actually against a specific pathogen. So it's got hands, but only for one guy. It has one op. That's it. So the macrophage cells that chomp up attackers, so dendritic cells, neutrophils, basophils, eosinophils, all under innate immunity because they just attack whatever's in sight. And what's really important here is that the macrophages actually offer you the bridge over somewhere because these can become, like we said before, antigen-presenting cells. What are antigen-presenting cells? When a macrophage chops up a bacteria into pieces, you can have little pieces of bacteria that present themselves on the surface of the macrophage, which we're going to talk about later. That's a tiny piece of the bacteria that it can be identified by. This macrophage that ate and chopped up the bacteria is now known as an antigen-presenting cell, because it's presenting an antigen. And this antigen-presenting cell is actually the bridge over to adaptive immunity. And this is where B and T cells against specific pathogens attack, and they recognize via antigens. They recognize via antigens. Does that make sense? Strange example. Have you guys ever had a vivid memory somewhere from childhood and you see it and you're just like, oh my God, I remember that place? Or you smell something, you're like, whoa, I remember this specific day of my life that I smelled that. That's almost like this. These guys see that antigen and then they're walking around the bloodstream. They're just like, whoa, I know that guy. Let's go get him. Right? Get him. Get him. Right? Where are white blood cells produced? In the bone marrow, where the rest of the blood cells are produced. And what is one of the big organs where white blood cells are stored? You got the lymph nodes and you got the spleen. Very good. So, lymph nodes are places where the immune cells communicate. It's possible that an antigen-presenting cell is presented to a B cell or a T cell, went over to a lymph node, told everyone else. Right? And now we're going to mount a response, and we're going to see what that response looks like. You guys buckled up? You guys ready? 1 second. Okay. Let's keep going. Let's leave this here, actually. Let's talk about the innate immune system. You have a first line of defense. So I'm going to continue on the innate side. You have a first line of defense. These are defensins. Literally just enzymes that can tear pathogens to shred. And they're found on your skin. You also have mucous membranes, which can trap pathogens. You also have hair in your nose that can trap pathogens. You also have a mucociliary escalator in your respiratory tract, which can send pathogens back up. And every time you go and swallow it, there's probably a little bit of bacteria in there that you're killing with your stomach acid. That brings me to the next thing. What about your stomach? What about all those bacteria that would have gone to your bloodstream but just got neutralized in your stomach? Right? And those mucous membranes contain that same enzyme we talked about in the last chapter, lysozyme, which just tears things to shreds, rips it apart. You'll see that the immune system is really violent. It likes to do things in a very dramatic way. It's almost like me. It's very aggressive, very violent, very forward, makes a statement. But it means well. I don't know if I mean well. Do I mean well, guys?
You want us to get a 476?
Huh? See me after class. You have... I have no brain cells is what I have. You have something known as the complement system. Has anyone here ever been to healthcare, like school before, where you've studied the immune system in depth? How confusing is complement, man? So confusing. Can someone anywhere, look up in your laptop, just look up the complement pathway. Just look it up. And just look at how confusing it is. C3b and C3bbb and C3a and C4d and C blah blah. And all of them are just named C. This has to be made up. Like when you look at it, it just has to be made. Just look at it. Just take a look at it. I want you to look at it and tell me that's not fake. There's no way. Look, come on. The complement system is a system of proteins that kill bacteria. That's it. Done. Don't need to know anything else. There's a classical pathway, an alternative pathway, and complement proteins, and they activate one another, and there's a membrane attack complex, and C5, and this and that. No. Proteins kill bacteria. Done. Does ever tie your hair? You had long hair before, right?
Yeah.
You tie your hair up and you start to get that like tight headache in the back.
Pulls it.
It's just not long enough. My hair's just not long enough for that.
Yeah, you have to tie it low to not get the
Yeah,
extra.
it's so annoying. What?
I said I thought hijabis don't have hair.
It's true. Interferon. Interferon is produced by virally infected cells. And this decreases local viral protein production, increases MHC1 and 2 expression, which we'll talk about in a second. And this also causes flu-like symptoms. SXS means symptoms. What do you want me to read? All of it. Great. Okay. So we have, these are all your first line defenses.
We just tried those. Yeah.
Oh, interferon. Produced by virally infected cells, right? Leads to decreased local viral protein production locally in that tissue bed, right? Increases MHC1 and MHC2 expression, which we'll talk about later. You look like you're having a great time. And causes flu-like symptoms. How much space do I have here?
Where your hand is like your thumb is blocked.
Oh, okay.
Yeah, your thumb is gone.
All right, cool. You guys done up here? And I'm just going to keep going here. You know what? No, I'm not. Bye-bye. So let's talk about some of the cells of the innate immune system. So let's start with macrophages. Macrophages. They are agranulocytes. Activated by the presence of a bacteria nearby. This activated macrophage will consume the bacteria via endocytosis. Remember, this is where we push the membrane in and make a vesicle and pop it into the cell. It will also chop up the bacteria and present antigenic meaning identifying, things that can identify the bacteria, basically a little ID card. ID in medicine means infectious disease, so I don't love putting that there, but whatever. Antigenic parts of the bacteria on its surface using major histocompatibility complex MHC proteins. Major histocompatibility complex. We now need to explain the difference between MHC1 and 2. This is a point of confusion for some students. I don't really know why, to be honest. I remember struggling a little bit when I was a student, but I've sort of synthesized it in a way that I think is very straightforward. I think we can make sense of it. One of them is an endogenous pathway. One of them is an exogenous pathway, meaning one of them is purely inside the cell. One of them starts outside the cell. Do you guys get that? Do you guys understand what I mean by that? So one of the pathways actually begins with a molecule that was found within the cell itself. Nothing from outside. I think that's the little bit of confusing part. How do you get an infection in the cell without anything from the outside? Let's talk about it. So let's do MHC2 first, because it's a little bit easier to understand. Because MHC2 is this one, right? And draw out some steps. This is a bacteria. This bacteria has a square antigen that you can identify it by. This bacteria will get eaten by a macrophage. That macrophage will chop the bacteria into pieces and preserve its antigen. There is a molecule inside the macrophage, known as MHC2. It is a stand. The antigen goes on the stand. And now you have, should have done the macrophage in a different color. Macrophage, stand, chopped up identifying antigen on the stand. On what stand? MHC2, major histocompatibility complex 2. Yes? That's the stand. That's the bacteria. And you can ID that bacteria using that stand. So now, the macrophage will actually take that stand and place it right here on the surface of the cell. Say, yoo-hoo. Come look at this. Look what I got. Look at this fish I just caught. Right? Macrophage. I'm going to drill this home. MHC2. Yes?
Yes.
Good. A CD4 plus T cell will come recognize that and pass on the information. ... And we'll talk about what happens after that. How are you going to remember all of this? 2 requires 2 environments, inside and outside. 2 requires 2 environments. If the antigen came from the outside, it gets chopped up and put on MHC2. How are you going to remember that it's CD4? There's something known as the rule of 8s. CD4 recognizes MHC2, which multiplies to 8. CD8 recognizes MHC1, which equals 8. CD4 T cells are known as helper T cells. Because the ... here actually doesn't lead to cell death yet. This leads to B cell activation, and that leads to antibody production. And that's the end of the line there. Because now you make antibodies against that bacteria, and you can go attack it and attach these antibodies, and antibodies signal for the rest of the body to kill that bacteria. Does that make sense? Bacteria in, macrophage eats from the outside, 2 different environments. MHC2 takes the antigen, presents it. CD4 passes the message along to B cells, makes antibodies, neutralizes the bacteria wherever it sees it. Yes?
Can you think of CD4 as a secondary messenger to a certain extent?
Kind of, but they have a lot of other jobs.
No,
so
of course, it amplifies the...
yes. Yeah.
Okay.
And there's a very complex process that actually goes on between the CD4 and B cell. So like this isn't just CD4 coming, doing the job, and then going away. The 3 of these things need to work together in some way. This is known as the exogenous pathway. Now, you guys ready for the other one? MHC1. MHC1 is a little more difficult to contextualize until you remember how, if you want to take a photo, take a photo. Until you remember how it starts. If you remember how everything starts, you're golden. Okay. Guys, how is there an antigen inside the cell that the cell did not pick up from outside? If you know the answer, please don't answer. Just think about it for a second. How did an antigen get inside the cell without the cell specifically picking it up? Someone put it there. Someone put the antigen in the cell. And the only things capable of doing that are viruses. We have a macrophage and a virus. And that virus injects its genetic material into the macrophage. Unknowingly. Unknown to the macrophage, is now infected. Yes? The macrophage then uses the viral DNA and replicates, transcribes, and translates, which will produce viral proteins. So now your macrophage that is infected, I'll put a star right here, is now producing viral protein. Note on MHC1. Every cell in the body has MHC1. And you'll see why. Because MHC1 checks to see, is the cell making anything that we don't recognize? Is the cell producing anything that we don't recognize inside of it? Right? These molecules we've formed get placed on another MHC, known as MHC1. I will draw it a little differently. This is MHC1. Say hello. I said say hello. The viral protein that we can consider the antigen, in this case, will be placed onto the MHC1 molecule within the macrophage. You guys have to learn some manners. We just met someone new. So we have our MHC1 molecule that is endogenous to every single cell in the body to check to see whether or not it is producing the right things that are needed within the body. And we put something unknown on top of it. Yes? This then leads to presentation of that antigen on the surface of the macrophage. A CD8 cell will then recognize this and signal for what? B cells? No, it actually signals for the absolute frickin' explosion of this cell. Kill it now, kill it right now. Because this cell has now been hijacked to produce something that the body is completely unaware of and completely unknown to the rest of the body. We have no idea what it's going to do, how it's going to do it, what this thing is. It's not us, and we don't want it. Blow that cell up right now. Because deep within that cell, which you probably forgot about, is still that viral DNA, which is capable of producing more viral protein and infecting the rest of the body. So when the CD8 cell comes and sees that this thing is infected with something that's triggering on MHC1 that isn't a protein that the cell makes itself, it blows that cell up. This is why CD8 T cells are called killer T cells, not helpers. Yes? It's actually injecting the cell with like self-destructive enzymes. It's a different pathway than both. I believe they're called granzymes. The CD8 cell comes, just punctures a hole in the cell, drops off a bunch of enzymes, and it just destroys from the inside out.
Is the MHC1 in the cell before the virus?
Yes.
So it's from the beginning.
Every cell is naturally born with MHC1 inside of it. Every cell in your body. Yes?
So you said for MHC2, it's the pathogen is a virus. Would it?
MHC2, the pathogen is a bacteria.
Okay, MHC2, it's a bacteria. For MHC1, it's a virus.
Yes. You guys can see my notes from 2021 when I was studying for the MCAT. And I was trying to make sense of what you guys are studying right now. I feel so old. Yes? We were just about to talk about that. There's another type of T cell called a natural killer cell. An NK cell. These cells have one specific job. Kill any cell that has a downregulated, decreased MHC1 expression / activity. Why is this important?
Because if the MHC receptors are down, that means it can't present, or like it can stay hidden within the cell.
Zaid says if the MHC1 is decreased, then you actually can't present the virus properly. It can stay hidden within the cell. That's absolutely correct. If you didn't have the MHC1 activity to show the viral protein over here, that virus is just going to replicate and replicate and replicate and replicate and go and go and go and go. What? And now you're not even doing the body's job anymore. You're doing the virus's job. Yeah? Very good.
So there's natural killer cells and cytotoxic. Okay.
Granulocytes? You guys good with that? We'll move on. Yeah.
Quick question. So you said that natural killer cells kill cells that have decreased MHC1 expression. When you're talking about interferons and how they have increased MHC1, like how does that work?
So these virally infected cells sometimes know that they're virally infected, and they'll just release interferon so that anything in the vicinity, remember I said local viral expression and local MHC1, it tells everything around it like, hey, I'm going to die. Like, I know I'm going to die. I know I'm virally infected, so someone's coming for me right now. But all of you guys should amp up your defenses a little bit locally.
Got it.
You guys good? Okay. So once again, we talked a little bit about the adaptive immunity. We talked a little bit about the B cells and the T cells, but mostly that was the innate immunity side of things, right? And we're still going on that. We're still talking about innate immunity. Yeah, see how stuff goes a little bit faster once you pass like chapter 5, chapter 6, right? Let's talk about the neutrophils, eosinophils, and basophils. Neutrophils can follow bacteria using chemotaxis. Bacteria leave a little chemical trail, and the neutrophil can follow that, right? And they are also able to eat bacteria, so they are phagocytic. Very medical term on the board there. Num num. And believe it or not, anyone here ever had like really bad acne before? Anyone? Yeah? And then like what comes out of your pimple? It's pus, right? Pus is what? A bunch of dead neutrophils. Pus equals dead neutrophils.
Wait, I have a question. Is MHC2 part of innate immune system?
Yeah, because it's part of macrophages. Question was, is MHC2 part of the innate immune system? Answer is yes. But once it signals over to the B cells and the T cells, now you're in the adaptive. And that was the sort of important thing to learn there, that the MHC allows for the communication between the adaptive and innate immune systems. So we could hunt down every single bacteria one by one with phagocytic cells. Like that's not something that we're incapable of doing, but that would take forever. And they're probably going to multiply faster than you can eat them. You need to recruit some help from the adaptive immune system, which we're going to take a look at in a little bit. Eosinophils. This is your histamine response. Increased vasodilation. That scares me every single time. Flushing. Allergy. Things like that. Eosinophils are also important for parasite infections, but that's not something they teach you on the MCAT. Parasites are pretty hard to deal with. You guys want to look up a cool parasitic infection? Look up Loa Loa. Loa, Loa Loa.
What's the last word that you wrote?
Allergy. Basophils are basically just special eosinophils. That's it.
Ew.
Loa Loa. Hey.
What's the point of vasodilation for the stains? Does it just like make them travel faster, or?
Do you guys want me to actually explain that? It's very interesting, but it's a little bit complicated. You guys want me to actually explain what the vasodilation does? When you have an infection, and a big part of the reason why people who are septic get massive vasodilation and drop in their blood pressure is because of one thing. Let's say you have a tube, and there's pressure in this tube. right? And you are flowing down this tube in a laminar fashion. All of a sudden, you hit a part of the tube that rapidly just expands. This is the vasodilated portion of the blood vessel. It is usually in the post-capillary venule. That's where vasodilation takes place. Am I in frame? Okay. As you come crashing towards here, boom, 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 you're going. Once you reach the point where the spout opens, what's going to happen? Because of your widening of the blood vessel and the fact that it widens out, it slows down, goes turbulent, and moves out to the edges. So everything whoosh, moves out to the edges. And interestingly enough, on the edge of the blood vessel, on this side of the blood vessel, you have all of the proteins that are able to pick up the neutrophils and the eosinophils and the basophils and all the infectious chemicals and blah, blah, and this and that. And so there's literally a neutrophil pattern called rolling, adhesion, transposition. So because the infection is in the space around the blood vessel, in the tissue bed, right? So the neutrophil hits the vasodilated segment of the vessel, rolls against the edge because of the turbulence and the widening of the vessel, which slows down the blood flow, right? Because you guys know the blood flow equation. I forgot it. It's related to the radius of the tube, right? The flow equation. Yes? Someone who studied physics, just say yes. Just say yes. Okay, good. So that is true, by the way. If you open up a tube more, the flow through that tube will slow down. That I know is true. I just forgot what the equation is. Slows down, neutrophil rolls, attaches to not an integrin. I forgot what the protein is called. Hold on. Attaches to a protein that catches neutrophils and flips over onto the opposite side of the blood vessel and goes to fight the infection. This is why vasodilation takes place. Does that make sense? Yes? Okay. I want to do adaptive immunity, but it's just such a bad idea. It's just such a bad idea right now because we have to pray, and it's also Sunday. And adaptive immunity only takes like 30 minutes. And then because next time on Thursday or Friday, are we on for Thursday this week? Okay. Because next time would be digestive, and then finally we're going to do the homeostasis lecture, which is going to take like an hour and a half. That's the kidney, the RAS, the skin, ADH, fluid balance, all of that all together. Digestive system takes like 30 minutes, literally. So it's up to you guys. We can pause, pray, come back, finish. Or we can do this in the first 30 minutes of next time and then do the digestive system, which is another 30 minutes, and then use an hour and a half for homeostasis. What do you guys want? You want to go home? You guys want to go home? Guys, thank you so much for watching. I'll see you next time. We're going to cover the rest of this in the next video. I knew this was going to be a part one. Let's go pray and let's go home. I'll see you next time.